Now look at this.
What's up, penguins? So today we're going to do topic 2.4 on the plasma membrane. So the main part of the plasma membrane is made up of course, phospholipids. And so it's a bilayer, so there's going to be 2 layers of phospholipids that will face one another. We also see a lot of proteins in it, so it makes this thing called the fluid mosaic model, because when you look down at it, you kind of see, number one, that it's very fluid, because the phospholipids are able to laterally move past one another. And then the mosaic comes from the fact that you've got all these little proteins kind of embedded in that protein that makes like a mosaic. You also have a lot of glycoproteins, and glycolipids. So there's going to be a little carbohydrate that's going to be attached to a protein or a lipid that can help for cell-cell communication and recognition of the cells. You're going to find that there's cholesterol found in the plasma membrane. That cholesterol is going to inhibit the phospholipids from getting too close together when it gets cold or too far apart when it gets warm. So we talked about the phospholipid before, right? So a phospholipid is made up of a phosphate group and 2 fatty acids. So it's going to associate into a bilayer where you've got the hydrophilic heads, the phosphate heads that are negatively charged, will face the interior of the cell and the exterior of the cell, with those hydrophobic fatty acid tails facing one another on the interior of that membrane. This causes the membrane to be nonpolar. So when we start talking about things moving across the membrane, we're going to have to remember that polar substances are going to be less likely to pass through this region because polar and nonpolar don't traditionally mix. So proteins are going to be embedded within that membrane. So it's important to know those 6 different functions. So it could be a transport protein. So it will transport materials from one side of the membrane to another side of the membrane. There's 2 different types. We'll hit those a lot more when we get into membrane transport, but it's channel protein and carrier proteins. They can also act as enzymes, speed up chemical reactions occurring on the membrane. We also have receptors, so they can bind to a ligand to cause some type of signal transduction pathway, which we'll talk about later. We can act as a cell surface marker. So this is for cell recognition to, recognize our own cells from pathogens and be able to have cells interact with one another. Cell adhesion, so holding 2 cells together, and then attachment to our cytoskeleton. So this gives that rigidity and support to different cells. So we mentioned before that cholesterol is important for our plasma membrane. So here you can kind of see what would happen in a cold environment without cholesterol. So it would be more rigid. It's not going to be able to be fluid. And so oftentimes our membrane might break. If it's too hot and we don't have cholesterol, the membrane becomes too fluid. It will get a little bit more flexible. There's all these gaps in the membrane. And so it won't hold its shape, which could be a problem for a lot of our cells. Remember, I'm doing this for every single one of the topics. So hit that follow button. Remember, AP Bio Penguins, dress success. Bye, y'all.